A new vaccine for multiple sclerosis. A recent press release from Pasithea Therapeutics suggested a successful study of a DNA vaccine in a mouse model of multiple sclerosis. I'll give my take on the study, followed by an interview of the chairman of Pasithea Therapeutics, Professor Lawrence Steinman. Let's have some fun. So before we get into the vaccine, I want to give a little background, which is the idea of molecular mimicry between two proteins, EBNA1 and gliocam. EBNA1 is one of the proteins of the Epstein-Barr virus known to be a potential cause of multiple sclerosis. Virtually everyone with multiple sclerosis has evidence of prior exposure to this virus. Separate video on this topic by clicking the card above. And it's known that in animal models of MS, EBNA1 exacerbates an MS-like disease known as experimental autoimmune encephalomyelitis. And many people with MS have antibodies against both EBNA1 and gliocam, and they can be cross-reactive. In other words, antibodies against EBNA1 can bind gliocam and vice versa. So what is gliocam anyway? It's also known as hepacam, and it's a protein similar to immunoglobulins or antibodies that's a cell adhesion molecule expressed in the glial cells or supportive cells of the nervous system. So let me introduce the concept of molecular mimicry, which is the idea that your immune system sees one antigen and develops an immune response to a different but similar antigen. So for example, in this diagram, you can see a T cell interacting with a foreign antigenic peptide, in other words, piece of the protein represented by different shapes here. And it's very similar to a self-antigen, say something in myelin basic protein or gliocam, and it accidentally induces an immune reaction against the body's own tissue. And this is very similar to how, say, a viral infection could trigger Guillain-Barré syndrome, a demyelinating disease of the peripheral nervous system. It's believed molecular mimicry could be part of the pathogenesis of MS. And if I look at the proteins myself, gliocam and EBNA1, they don't look that similar, but according to one author, the C-terminal portion of the intracellular domain of gliocam is similar to an EBNA1 domain previously linked to risk of MS in a prior basic science study. And indeed, you can see in the middle of the proteins, there is a very similar amino acid sequence that overlaps almost perfectly, which could be driving molecular mimicry and could be part of the cause of MS. And gliocam ends up being a very important protein in myelin function. It's known to increase cell adhesion, the binding of cells of the central nervous system together, and motility, movement of the cells, and it also regulates cellular traffic of a different protein called MLC1, which is part of the cause of a genetic demyelinating disease called megalencephalic leukoencephalopathy with subcortical cysts, or MLC1, and mutations of gliocam, in addition to mutations of MLC1, can cause this condition. And just to look at some MRI scans of 3 individuals with this disease, you're seeing A, B, and C is a 3-year-old, D, E, and F is a 6-year-old, G, H, and I is a 10-month-old, and you can see these extensive subcortical and periventricular demyelinating lesions. They don't look like MS, but definitely this protein is very important in myelin function. And targeting a protein homologous to a protein in Epstein-Barr virus makes a lot of sense because of the extremely strong evidence that EBV, the cause of mono or glandular fever, is part of the cause of MS. For example, this study from the US military compared people who were EBV positive, who had antibodies against the virus, to those who were negative. Those who initially tested positive were 26-fold more likely to get MS, and those who seroconverted, in other words, those who got exposed to the virus later, were 32-fold more likely to get MS than those who were negative. And if you looked at people who were initially negative for Epstein-Barr virus, who did not have antibodies, but went on to develop multiple sclerosis later, 34 out of 35, virtually all of them, tested positive for antibodies before developing symptoms of MS, suggesting that Epstein-Barr virus infection is necessary to develop MS. What happened with that one person? Well, no laboratory test is 100% accurate. So finally, we move to the vaccine. This product has a preliminary name of PAS002, and it's developed by the pharmaceutical company Pasithea Therapeutics. And it's a DNA tolerizing point vaccine. And it uses a circular form of DNA known as a plasmid. And so the DNA enters your own cell and uses your own cell's machinery to transcribe and translate into the protein GlialCam-1, kind of similar to the technology of messenger RNA vaccines used for COVID-19, the Moderna and Pfizer vaccine. And it's thought to work as a reverse vaccine. Instead of boosting your immune system, it induces immune tolerance, kind of tamping down your immune system's reactivity against GlialCam. Now, you may not have heard of this drug company, Pasithea Therapeutics, and that's because they don't have any approved products. It's a very small company with a market capitalization of only $35 million and currently no revenue. I'm not encouraging gambling here. And this is the information from the press release of the animal study. This is very preliminary. This is an animal experimental autoimmune encephalomyelitis study. Mice are not humans. Mice do not get multiple sclerosis, but this is where the process starts. So what they did is they induced this disease similar to multiple sclerosis in mice, called experimental autoimmune encephalomyelitis. Then they gave the vaccine intramuscularly at days 0, 3, 7, 10, and 14. 0 is with respects to the induction of the disease. And of course, they compared it against placebo. And they found that the drug delayed onset of paralysis when compared to placebo, p less than 0.001. It reduced disease severity, p equals 0.002, and prevented any disease whatsoever in roughly 50% of the mice, p equals 0.004, all highly statistically significant. Again, this is very preliminary data. A few miscellaneous topics. GlialCam is also in the monkeypox virus, so there's some potential use as a monkeypox vaccine, and a provisional patent has been filed. Of course, all this data is not published, but will be published and presented at professional conferences fairly soon. And now we hear it from the horse's mouth himself. I have Professor Lawrence Steinman, who is the chairman of Pasithea Therapeutics, and he's a distinguished professor of neurology. He's done a lot of great research on multiple sclerosis, well known for his research on integrins and helping with the development of the drug Tysabri. Thank you for taking the time to do the interview.
My pleasure to do this.
So there are 1,000 projects you could have worked on. What inspired you to get involved with a potential vaccine to treat multiple sclerosis?
Well, I've been working on this for more than 20 years, and an earlier version of this approach went into a 267 patient phase 2 trial. The idea is that if we could find out what triggers multiple sclerosis, and there's been a very big breakthrough that was published this winter, if we could simply remove our immune system from attacking whatever is triggering the disease and leave the rest of our immune system intact to fight COVID, to fight shingles, to fight the common cold, we would be in good shape. The medicines that we use now for multiple sclerosis, many of them have a high impact on what we would call the normal functioning of the immune system.
All right, we've certainly learned that the hard way with COVID. I probably personally have over 100 patients who have been hospitalized with COVID, many taking immunosuppressant drugs, and I'm sure you have a similar experience.
That's right. So we would like to know if we could just shut off the response to what's unwanted and what leads to multiple sclerosis and leave the rest of our immune system intact to fight viruses and bacteria. That would be ideal. And this winter, there were 2 papers that came out. The first was from the Harvard School of Public Health, and they studied 10 million military from the US military, and they evaluated blood samples, and they found that 801 individuals in this big cohort from the military had multiple sclerosis. 800 of them had positive antibodies in their blood to the Epstein-Barr virus, the virus that causes mononucleosis and some other things. And they also had 35 patients who entered the military and were negative for any antibody to EBV, but 34 of the 35 became positive before getting MS. So Alberto Escarrillo at the Harvard School of Public Health showed that the risk factor for testing positive is 32-fold. But the data that came out that I just mentioned were so compelling that a wonderful epidemiologist actually used the word cause. And that's very rare. We can say that on a few occasions. Smoking can cause cancer, and Epstein-Barr virus can cause multiple sclerosis. I put the can in there because the first thing that people would ask is, the Epstein-Barr virus, somebody will say they'd read somewhere in the Escarrillo paper and elsewhere that more than 95% of us who don't have multiple sclerosis test positive for the Epstein-Barr virus. So the way I look at it is the Epstein-Barr virus can trigger MS, but it's not sufficient to cause MS in almost all of us, that there have to be other factors that have to come into play. And I know some of these other factors, but let me tell you the other paper that came out a few days after the Escarrillo paper. So the Escarrillo paper was in the journal Science, and the paper from my group at Stanford was in the journal Nature. And, and working independently, we had been studying the antibodies that are found in the spinal fluid of MS patients. So these are antibodies that are almost monoclonal. They're produced by one expanded group of antibody-making cells. So we know some of those antibodies. Some are directed to measles, some are directed to rubella, some are directed to the chickenpox virus. But we found a big population of antibodies that were directed to the Epstein-Barr virus and to one particular piece, a piece called Epstein-Barr nuclear antigen 1, EBNA1. And then we did something that is enabled by modern technology. We were able to ask, is there something in the human proteome of all the 16 to 20,000 proteins that we humans make, that monoclonal antibody that bound to a virus, the Epstein-Barr virus, may have also bound to something within us. And we found a molecule that the clonal antibody binds to called glial-cam, glial meaning glue, and cam, the abbreviation for cell adhesion molecule. So the glial-cam and the Epstein-Barr nuclear antigen 1 share a certain stretch that's nearly identical from one to the other. And that gave an enormous clue. There's an animal model of multiple sclerosis that's widely used. When we inject, you can't infect an animal, a rodent with Epstein-Barr virus, but you can give them an injection of gliacam, and that caused pretty severe paralysis. Then we looked in humans, and at any one time in their blood, not in their spinal fluid, about 25 to 30% of patients have antibodies to glialcam. We have to do, and we're doing the longitudinal study to see how predominant that is in the spinal fluid, but it gave us a very big clue. So the idea here is, if Epstein-Barr virus is the trigger, and if an unwanted immune response to this piece of the virus that resembles glialcam is the big culprit, would it be possible to make an inverse vaccine, to leave everything intact, but to shut down the person's ability to make an immune response to glialcam? So that's what we reported and what we're very excited about at this small company called Pasithea. Now, the experience goes back a long time. Back in the year 2000, we started another biotechnology company that took this pretty far. We used as a platform for making what I call an inverse vaccine. A normal vaccine is, you get a polio vaccine, you're immune to polio. You get a tetanus shot, you're immune to tetanus. That's conventional vaccine, and it's wonderful. We're trying to do something that never has been taken all the way to an approved product. Many individuals have tried. The idea is to make a vaccine that would tweak the immune system so it will not respond to just one or two things. And the thing we want it not to respond to is GliaCam. So when we started this activity about 20 years ago, our platform was DNA, and we engineered the DNA so that it would express what we want to. And we put some bells and whistles into the DNA so that instead of activating the immune response, it would turn down the immune response, but only to what that DNA was encoding. So the DNA has about 3,000 pieces, and one of the smaller pieces is set up to code for the protein we want to suppress the immune response, to make it an inverse vaccine. In the rest of the DNA, it's called non-coding DNA. It does not code for a protein. It used to be called junk DNA, but it's not junk. It contains many signals that tell the immune system to rev itself up. And one of the signals is a sequence called a CpG nucleotide. It's a short nucleotide. So back in the days when we first ran this experiment, we removed some of the CpGs, and we didn't know about GliaCam. So we tried to see if we could suppress an immune response to another myelin protein that we know is attacked in MS called myelin basic protein. It happens to be the most prevalent protein in white matter. And we ran the phase 1 trial and showed safety. And then in a big phase 2 trial involving 267 patients, and we published it in a solid neurology journal, Annals of Neurology, in 2008, we showed that we could reduce MRI activity in this classical type of phase 2 trial. We reduced MRI activity by about 50%, but the p-value in the compared to the placebo group was 0.07. So 0.05 is the magical cutoff. We had other evidence that there was activity with some of the secondary endpoints, looking at different ways of looking at the MRI. And it would have been sufficient to actually continue the development, but some other good drugs had come out at that time. One was Natalizumab, the anti-integrin drug that was just much more potent than what we could do with antigen-specific tolerance. And another was Fingolimod, an oral drug that again is reduces relapses by about 50% in the phase 3 trials. And we looked like we were below that threshold. So we understood, though, that drugs like Natalizumab have problems. It can cause a fatal neurologic disease. And at one point, it was withdrawn from the market. It returned, and about 250,000 people in the US have taken it, and it's a successful drug, but it does carry a serious warning. And I'm mentioning this because the type of thing we want to develop with an inverse vaccine is something that would be perfect. You get benefit from stopping to make an immune response against something you don't want, but you don't face the risk of something terrible, as we saw with the development of Natalizumab or the development of Fingolimod. There, they have a problem with certain kinds of herpes infections. So fast forward from when we discontinued work on the DNA vaccine about 10 years ago, we received funding for the work at Pasithea, and we decided to go for it again, but to engineer the DNA backbone so that it had none of those CpGs. Now, Brandon, an interesting thing happened. that about a year ago, a company called BioNTech, a slightly bigger biotechnology company, most famous for what we call the Pfizer vaccine. It's really the BioNTech Pfizer vaccine. And they used RNA to make a brilliant vaccine. Moderna did a similar thing. And those are the two vaccines that changed everyone's life in the direction of normalcy again. I say in the direction, they're not perfect, but they certainly keep about 90+% of people who used to be hospitalized for COVID out of the hospital, old folks like me. And it's been a great success. So the BioNTech people published a paper in January of 2021 showing that they could also do an inverse vaccine by tweaking the RNA technology so that instead of making a brilliant conventional vaccine, it's hard to say the sentence RNA and conventional because it's all so new, but a vaccine that does what it's supposed to do. We don't want to get infected with COVID. We want to be able to fight back hard. So they made a really good vaccine against spike protein, and it really is protective. So they made a tweak so that if they want to, they can shut down an immune response to an antigenic stimulus that is unwanted. Obviously, we do not want to shut down a response to spike protein. We want to make sure it's there, that it persists, and that it's good enough to, every time the spike mutates a little, we still can handle that. But this is a different world to try to make something that no one has ever taken successfully across the finish line to do an inverse vaccine. So we're very persistent and bullish about seeing how far we can go. It is somewhat, I have a smile on my face to say that one of our main competitors is BioNTech with their RNA vaccine. So all I can say is we have had some experience in the clinic with our DNA vaccine in humans. To my knowledge, at this moment, BioNTech has not yet advanced to humans. And now we're going to try to do what we attempted 10 years ago in MS patients, but this time I think we'll be doing it with the correct antigen. Instead of myelin basic protein, we'll be trying it with glycan. So the experimental results so far look really good, and we're going to be advancing those, publishing along the way, and we want to bring this into the clinic. So in conventional vaccines, so far, RNA vaccines seem to work a lot better than DNA vaccines. We talk about what vaccine did a person get, and the BioNTech, Pfizer, and Moderna vaccines are the dominant force. There were some DNA vaccines, they did not compete as well in terms of their testing. That doesn't mean that for inverse vaccines that we may see that DNA not as strong a conventional vaccine. It may provide a platform that's very strong for an inverse vaccine. Again, with all medical science, early stage, it will be experimental results that determine the outcome. I would be happy if they both work very well. And I think individuals, more importantly, who have diseases like MS would be even happier than the scientists. But that's what we're attempting right now.
Well, competition is good, and hopefully one or both will prevail. Now, to be a little bit technical, let's say I get the vaccine, my own cells transcribe and translate it into gliocam. How does my immune system know to be tolerant to gliocam and EBNA1 rather than exacerbate the immune response?
Right. So with most vaccines, we don't just give the antigen and that's it. We give something to make the immune system rev up. So with, let's say, the conventional tetanus shot, the tetanus toxoid is in alum. It's an adjuvant which intensifies the immune response. The immune system sees a foreign material, alum, and the tetanus, and that double signal, this two-step signal, tells the immune system to rev up. So here we're presenting the antigen, but in the absence of this CpG stimulus. So the antigen alone is able to shut down the immune response rather than rev it up. And it's the absence of giving an antigen in the absence of a danger signal. Look, gliacam is all over the brain. A little bit of it is in the liver, and we normally don't, 99% of us don't have immunity to it, even though we have evidence of EBV. EBV elicits an immune response because it's a virus and it has a lot of other components which give it a danger signal. So we're presenting the antigen in a format that is generally considered self, leave me alone, this is not a danger. And many people raise the interesting question, you raised the question, well, aren't you going to turn off an immune response to that little piece of the Epstein-Barr virus? And the answer to that is yes, but the Epstein-Barr virus has hundreds, if not thousands, of other pieces where our immune system can attack it. So we should be able to get rid of EBV, or at least contain it, as almost all of us do, and not have that unwanted reaction to that little piece. So that's what we're doing. And this has never, as I said, been successfully taken across the finish line as an approved drug. So there's a lot to learn. We may be poised at some kind of knife edge that can go one way or the other. So a lot of people will ask, what keeps you up at night about an inverse vaccine? And we haven't seen it yet, fortunately, and maybe we'll never see it. What keeps me up at night is, are we missing something, or in some person, will it cause an exacerbation of the disease rather than a protection against the disease? So while I'm saying all of this, Moderna and some other companies are planning it, but Moderna is already in a clinical trial of a conventional vaccine to EBV. Maybe if they succeed, we won't see anyone get mononucleosis anymore. But one of the worries about a vaccine against EBV, might it worsen MS in people who already have it? And might it cause MS in people who otherwise were not going to get it? Those are open questions. I hope we don't see that. Everyone's going to be watching it very, very, very carefully to make sure that those undesirable side effects are not seen.
Yeah, I mean, I saw some of the seroprevalence studies for EBV, and even by age 5, there's a decent seroprevalence. So for the EBV vaccine, we probably have to intervene fairly early. But this is, of course, intended as a treatment of multiple sclerosis rather than as a prevention.
Right. Yeah, and you raise another interesting point. So if in 90+% of us, we have positive serology, is it possible that this virus actually does some good for us, that our B cells need, the virus infects the cells that eventually are in the cell lineage that leads to production of antibodies. Do we actually, is it providing some beneficial function? We know so little about our virome. We now talk about the microbial floras, the microbiome, there's the virome. There are probably a lot of viruses that do good. We know some that are harmful, that are cancer viruses. We know the Epstein-Barr virus can cause cancer in some individuals. It can cause mononucleosis in a fair number of kids that we see at the health service at Stanford, who it's spread by kissing. And as we know that if you're a young college student, there's a lot of kissing, and that's something good. When the 5-year-olds are getting it, they're probably getting kissed by mom and dad and grandparents and brothers and sisters. So that's how they get that seroprevalence very early on. So there's a lot to learn here. But one of the things that gives me comfort as we do this medical experiment is we have taken it into a few hundred patients. And so far, we've seen hints of a benefit. But the good news is we've seen so far that we've done no harm. And that's, of course, what we want to make sure of.
Yeah,
That's definitely a good sign.
Now, when you're talking about that prior vaccine trial, is that the study that Leslie P. Weiner, one of my former mentors at USC, was involved in, or is that a separate trial?
That's a separate thing. First of all, I have great respect for Les. I knew him well. He was chair at USC. Terrific guy. I enjoy him. And he had a trial. It was a vaccine against the T cells that might be involved. And there are about half a dozen companies that have different, very clever plans. Les Weiner's vaccine trial was one of them. It didn't get as far along as what I was telling you with our 267 patient trial, but just yesterday, a foundation that works on neuromyelitis optica told me about another cool technology that they're looking at. And, I think there's going to be many ways to solve the problem. It's very interesting. In 2022, we have no approved inverse vaccines. And maybe by 2030, we'll have 10 of them, and people can line up and decide what size is best for them, what type is best for them. But it's one of the goals in medicine to make specific immune suppression so that we don't have to take steroids or kill a big percentage of our B cells or trap our lymphoid cells and lymph nodes with sphingosine receptor modulators. Because all of those types of therapies, I refer to them as big hammers. We want to have a very precise scalpel for just excising that one or two immune responses that could be at the root of the disease.
Yeah, I mean,
when I talk to patients about like B-cell depleters,
rituximab, Ocrevus, I tell them it could be 0.001% of your B cells that are causing any problem, and we just don't have a way to target them specifically. So definitely a big hammer.
That's right. And with the big hammers, you're going to have some side effects that are undesirable. So I think everyone would agree that if somebody could succeed in making that scalpel that just hits what we don't want the immune system to attack, it would be a huge advance. Until then, with all thankfulness, we have some very good solutions to really bad diseases like MS with these big hammers. You have to be practical, and many people have much better lives because of the anti-CD20s that hit the B cells and natalizumab. People who get an acute attack take steroids for a short time to help attenuate it, and all of these things are very helpful. So we have to be practical, and maybe we'll never achieve that long sought after goal of antigen-specific tolerance or an inverse vaccine. Maybe we will. I think we have to keep trying, but I think we have to be very thankful as a medical community that there are good therapies, because when I started neurology, I trained in the late 1970s. We didn't have much, and now we have really a good arsenal of their powerful drugs. In my competitive mind, I like to call them big hammers, but they sure work. And, generally people do very well with them.
Certainly
the game has changed even since I've been in this field.
And I can only imagine the difference between 1970 and when I first started. It was probably enormous. But anyway, what is the next step for Pasithea? Are you planning on doing a phase 1 safety trial in people with MS? How far along are we from actually getting into human studies?
Right. We know the plan, the idea is we won't be able to totally piggyback on what we did 10 years ago, because as I said, we engineered out more of the CPG motifs, all of them to be precise, compared to just a few of them before. So I imagine the regulatory agencies would say, please test it in a standard phase 1 format. We would go into patients. I couldn't put this into normal individuals or people without MS. So within 12 to 18 months, we hope to have this. We'll have the toxicology done and all of the standard steps. We'll go to the FDA and we'll begin clinical testing. But the good news is, even though we have made some changes, there's at least some foundation for saying that this isn't completely unknown. DNA vaccines have been into people with MS. And I don't know what the timeline is for the competitors with the RNA vaccines.
Yeah,
I mean, obviously, no one can say exactly what will happen.
But not too far away from potential early human trials is the general point.
That's right. Yes.
Yeah, so we've got a lot of information. I appreciate your time. Any other comments about Pasithea, the DNA vaccine, or MS in general?
No, it's been a pleasure to work with the team at Pasithea, and we have other cool things that we're doing for motor neuron disease, which is another unmet need. We have a different integrin that we're trying to target in that disease. And the third disease we're working on is schizophrenia, where, again, the immune system may play a role, because one of the biggest hits in genetic analysis is a part of the immune system called the complement cascade, and it's complement C4A. So we're trying to make a molecule that will modulate C4A in the brain. So one of the things that is exciting about Pasithea, you talk about moonshots. We're doing antigen-specific tolerance against gliacam for MS. We're working on motor neuron disease with an approach that's been successful in MS with anti-integrins, but now we're applying it to ALS, and then in psychiatry with targeting the complement system. There's one other thing I wanted to say, and this is sort of, you're saying this at the last minute, so yes, I am. The pox viruses, including monkeypox, have a fragment in it that looks very much like gliacam. And the animal model that has been used to test many of these drugs, the experimental encephalomyelitis model, was originally developed at the Rockefeller Institute 90 years ago, because they wanted to know, why does smallpox and smallpox vaccine cause encephalomyelitis? And they gave us this model, and now we find out in 2022 that the pox viruses, including the virus that causes smallpox, has this piece of gliacam in it. That, when I first was told about that by colleagues, I nearly fell out of my chair. And that paper is under final review, and hopefully it will come out soon. But stay tuned for a lot of further stories about gliacam and viruses.
Yeah, so
Pasithea is definitely the
pharma pharmaceutical companies and going for the deep down, risking the interception, but hopefully some of these will come to fruition. Again, thanks for taking the time. I have some show notes with references below, and I'll update them if there is, in fact, a phase 1 trial to enter in the future. I almost had to cancel on you because I'm on call for jury duty this week, but it worked out perfectly. Thanks again for taking the time.
Thanks, Brandon. Wonderful to talk to you. And keep going at Kaiser. You guys do great things.
Take care.
Take care.
Bye.